Hello, I am Srividya Raman, a scientist at Cellular Technology Limited. Today, I would like to talk to you about a new imaging platform that we have developed at Cellular Technology Limited. This is the high throughput live dead yeast counting imaging system. Before I begin, I would like to take this opportunity to tell you a little bit about Cellular Technology Limited. We are a global biotechnology company that works towards developing new and customizable kits or platforms, mostly geared towards immune assays. We have a worldwide presence and more than 2,000 instruments that are being used by scientists and researchers in the immunology community. Our primary academic expertise is in immunology, and we were the first companies to have ever developed a microtiter plate-based imaging system. For the immune monitoring assays that I mentioned, we develop and validate experiments and protocols and methods to measure the amount of cytokines secreted by T cells. Now, these T cells are the primary defense cells in our body, whose activation is determined by the cytokine secretion, which then leads to an active immune response. Sometimes T cells can release multiple cytokines, and to detect that, we developed multicolor assays. These assays can be used either in a fluorescent-based system or in enzymatic ones. As we developed these assays, we noticed a linear correlation between the number of cells to the amount of cytokine detection. That was not surprising, but this then led to the determination of purity of these cells, and we developed that through viability studies that we developed at CTL. Now, these viability studies determine live, dead, and apoptotic cell numbers. However, as we furthermore, to quantify the secretion and cell numbers and such, we developed a device for detecting cellular products. This device eliminates any user bias, introduces objectivity, and improves reproducibility. These analyzers support professional GLP-compliant work. We have worked with many large corporations, such as NIH, and we understand the need for quality and regulatory systems that are in place. Our analyzers come with a highly developed, streamlined QC module which retains complete audit trails, including the tamper-proof archiving of original digital images, along with counting results. This also enables CFR Part 11 compliant work. In the recent past, we have been noticing a trend that yeast cells have undertaken from being used in the baking and brewing industries to now being used as model organisms for the pharmaceutical industries. These are workhorses for protein production. Since at CTL we had already developed a device and the assays to determine viability of mammalian cells, we decided to adopt the same technology to determine the viability of yeast cells. Now, the major challenge here was the consideration to size. Yeast cells are about 1/3 the size of mammalian cells. But I'm happy to inform you today that after a year of research and modifying and adopting our technology, we finally have developed a method for the detection of single yeast cells. But before I tell you about CTL's imager, I would like to tell you about what's available in the market today. Currently, to determine yeast viability, scientists perform colony detection methods. Cells are plated on a bed of agar, and colonies are allowed to develop in a controlled incubator. A couple days later, the number of colonies developed on the plates is counted by eye. This method is not just tedious, but is also subject to user bias. The other method is to detect cell lysis by measuring single-stranded DNA in the supernatant culture. This inconsistent and at best an indirect method of determining viability. The detection of single yeast cells can be analyzed by fluorescent-based imaging and counting. Cells are stained with a fluorescence-emitting dye, and the detection of fluorescence is a direct measurement of the cells. Membrane permeability can then determine live versus dead cells. Therefore, a two-color detection scheme would be ideal to detect viability. Now, there are multiple ways. Let's look at a few of them. The hemocytometer-based counting is easy to use. It has low maintenance, but it does not provide accurate counts over time, and this is definitely not high throughput suitable. The hemocytometer with automated counting, however, has distinct advantages over the manual counting method in terms of producing consistent results. However, the time for operation is long, as only 2 samples can be analyzed at any given time, and each slide takes about 1 minute to complete the scanning and counting. When computing hundreds of samples, this can add up. The flow-based counting system, on the other hand, is complex. It requires trained personnel to carry out the operations, and it needs constant maintenance. And of course, with the single-stranded DNA detection in the supernatant, it is easy to perform, but is indirect, inconsistent, and oftentimes it detects other nucleic acid components as well. The CTL Yeast Imaging Platform consists of the device, or the analyzer for determining cell counts, and the viability staining kit containing all the necessary reagents. With this system, you can easily image, count, live and dead yeast cells using fluorescent dyes. This is performed in a 96-well format, introducing high throughput analysis, and no transfers are required as cells are counted in the wells itself. This system can be scaled up to scan multiple plates serially using either a stacker or a robotic arm. This data can then be exported to any database of choice. In a little bit more detail, what looks like a box with a monitor is the yeast cell imager. This is a tabletop device of 29 inches in height, 28 inches in width, and 17 in depth. Inside this is a plate holder, and we can image multiple different plate formats, as we've done that with other assays. But for the yeast viability purpose, we have standardized the 96-well plate format. The analyzer has some complex optical components that allow fluorescent detection of 2 colors for yeast viability, but this can be expanded up to 8 colors. This is also able to detect images under white light in case you wanted to do some colony imaging. The resolution of yeast cells is about 12 to 16 pixels per area, and this allows accurate counting of cells versus clusters of cells. The images are scanned, analyzed, and counted automatically, thus introducing objectivity. The viability staining kit consists of low fluorescent plates. We tested multiple different plates to then determine one with low background fluorescence, low affinity to the dyes, and one that provides even distribution of cells on the surface of the well. We tested many different dyes to determine the ones that can stain live and dead cells that can be detected unambiguously in a mixed population. We took extra attention to make sure that these dyes were also non-carcinogenic. Given the size of the yeast cells, they are about 3 to 4 microns. We also modified the wash buffer so that no component in the buffer could be stained with these dyes, as it would be very difficult to distinguish debris from dead cells, thus eliminating any false positives. We also provide a humidified chamber in case you wish to run some proliferation studies. We'll talk about the proliferation studies later on. The workflow that we have, has been developed to be extremely simple. We start off with cells that are being incubated in a 96-well plate with whatever drug of choice as per your protocol. Now, this need not be a 96-well plate. It could be any reactive format. Here we mention 96-well plates as transfer of samples can then be performed with a robot or a multi-channel pipette. The CTL yeast dye is then added to these samples. The live cells are stained green and the dead ones are stained red according to the principles of these dyes. This is really quick and it requires no incubation time. The cell dye mix is then transferred to a 96-well filter plate. As I mentioned earlier, this low fluorescent filter plate is provided by us. The media is then filtered through using a vacuum filtration device, and the cells distribute on the surface. This is now in one plane, in a 2D plane, and can be imaged. We used the Pichia pastoris strain for our studies, and we cultured it in BMMY media prepared in-house. The live cells were used from culture, whereas the dead cells, we treated live cells with 95% ethanol for 1 hour to obtain the dead cells. We added the CTL-YD as shown in the workflow. At this point, I would like to mention that the CTL-YD consists of two components, the CTL-YD-a component and the CTL-YD-b component, and I will talk about this in a little bit. Live and dead cells were analyzed separately, and they were analyzed when mixed together in defined ratios. As I mentioned earlier, the CTL-YD has two components. The CTL-YD-a component stains dead cells. So in order to look at that in more detail, we took dead cells and stained them with this component alone. The images were analyzed with two different filters. Now, these filters are used to detect the emissions from the fluorescent dyes. One filter is suitable for the detection of live cells, and the other one is suitable for the detection of dead cells. When dead cells were stained with the dye and images were viewed, we saw a distinct signal from the image captured with the filter suitable for the dead cell detection, and we saw no signal leaching into the filter suitable for live cell detection. The same was true when we stained live cells with the CTL-YD component B, which is used for the detection of live cells. Again, we observed distinct signal from the image captured with the filter suitable for live cell detection, and no signal leaching into the filter suitable for the detection of dead cells. We can image both live and dead cells unambiguously with the use of these two filters. However, we were curious to know if we could image them simultaneously. Now that makes more practical sense, as most yeast samples will have a mixed population of both live and dead cells. We stained mixed population of cells with the CTL-YD dye. Now this dye, as I mentioned earlier, has both the components. And the ratio with which these two components have been added have been tested out extensively by us in the lab. We show you here overlays of the images with the two filters. We can see a 100% live cell population that's mostly stained with the live dye, a 50% live and a 50% dead cell population with separate live and dead cell staining, and a 100% dead cell population that is stained mostly with the dead cell dye. We can see both populations can be imaged simultaneously with the CTL-YD dye. However, to determine the safety, we mix different ratios of live and dead cells together and stain them with the CTL-YD dye as per the workflow, and the counts were computed by the software. As we can see, the percentage of live and dead cells computed correlate very closely to the percentage of dead cells and live cells added to these samples. Having looked at the determination of cell viability, a few other questions loomed in the development of a feasible viability assay, and we went ahead to address them. One of them was to determine whether the whole surface had to be imaged, or if imaging a portion of the well would suffice. To answer that, we stained a 100% live population of yeast cells with the CTL-YD dye and captured whole well views and a zoomed-in portion of the well. If you look on the left, the whole well view seemed to suggest an even distribution of cells in the well surface, but was unable to provide much quantitative information due to the lack of resolution. When we zoom into the image, it provides much better resolution, allowing accurate counting parameters to be set, so that single cells can be distinguished from clusters. This led to the next question, to whether the whole surface had to be imaged in a zoomed-in view, which would then impact time, or if we could just image one of these zoomed-in views. Therefore, to verify that, these cells were distributed evenly on the to verify if the cells were distributed evenly on the surface, different yeast dilutions, and we took 8 replicates each for all of these samples, and we imaged 5 different segments of the well: the top left, the top right, the bottom left, the bottom right, and the middle portion. The counts were then compared within each of these segments. We observed a consistent cell count from each of these segments of the wells. Also, the cells exhibited a similar trend corresponding to the actual number of cells per well. This eliminates any intra-well or inter-well differences in cell numbers. Another important was to determine the linear range of cell counts that can be counted on the 96 well surface. We determined this number to be 650,000 cells per ml, before which the cell numbers saturate out, but that's mostly due to the lack of surface area beyond that point. We know that no counting assay is truly validated without a comparison to the gold standard. In this case, that is the hemocytometer. So we took 3 replicates of live cells and we stained it with the CTL-YD dye and used a hemocytometer-based counting system to determine the cell counts, and we compared that to our own CTL's automated counting system. As you can see, the cell numbers are very similar to each other. However, the standard deviation with the CTL's counting system is tighter. Now that's not surprising given that manual counting is subject to user bias, and given the size of these cells, as I mentioned earlier, they were about 3 to 4 microns. That poses a challenge to the eyes. Now that is eliminated with the CTL's Yeast Imaging Platform. With the CTL Yeast Viability Platform, we employ an easy and a fast workflow. We have almost no equipment maintenance required. We can accurately and unambiguously detect live and dead yeast cells. We can sensitively detect any population of live or dead cells in a mixed sample. We have shown immense intra-well and inter-well precision with cell numbers. The scans and counts are performed automatically using the inbuilt CTL software, with which the time to scan and count each plate is now reduced to less than 15 minutes or less than 10 seconds per sample. Now moving to a few optional assays. We talked a little about colony formation and the detection of colonies as a means of determining viability of yeast samples. Now, single cell analysis provides viability information at that particular point in time. However, if you're looking for a replicative potential of these viable cells, you need to do colony assays. Therefore, we performed some similar assays with the CTL yeast imager. In this case, yeast cells were imaged as before, and following that, they were allowed to incubate with minimum media on the surface in a humidified chamber. If you remember, this chamber is provided by us with the kit. So the cells were incubated for 3 days, following which the wells were again stained with the dye and imaged as previously described. As we can see, the colonies that were formed on the surface can be scanned and counted automatically. Optional colony assessment assay is to seed yeast cells on a slab of agar, which is what is traditionally done. And then these are then imaged under white light. We have seen that we can count more than 500 colonies in the area of a 96 well surface. If you're interested for visual effects, we can create a dark field to provide better contrast between the colonies and the agar background, as you can see in the dark image below. A lot of times, researchers are just interested to look at turbidity assessment. The more turbid a sample is one that has proliferated more, and a clear sample is one that has not proliferated much. This is a crude method to eliminate a large sample batch. Here, we can use an adapter that forms a grid at the bottom of these wells, and the software images the grid to assess the turbidity of these samples. Now that can also be performed with the CTL yeast imager. And I've been talking a lot about our system being high throughput suitable. And these are some optional instrumentation that we can use to achieve that. The stacker on the right can hold up to 50 plates, and the robotic arm on the left can hold up to 240 plates, depending on the sample size that you would use. These instruments can be attached to the analyzer, and these instruments would then load, scan, image, and compute the data for each of these plates serially with no further input from the user. The stacker finds the first plate and loads it onto the plate loading region of the analyzer. The plate is inserted and positioned within. And the wells are scanned and counted in real time. What we are seeing here is a colony assessment assay. And as you can see, the scanner is going from well to well. Once it has scanned all of the different samples in all the 96 different wells, it ejects the plate out, at which point the stacker then picks it up and places it back to the original position that it came from. At this point, the stacker then looks for the next plate and it loads it onto the analyzer. The barcode on each of these plates is scanned and the data is associated with the barcode for easy access at a later time. We will be happy to come over and present the system to you. We provide training, customer support, and we do this globally, and we will be happy to do so for you too. You're welcome to visit our website www.immunospot.com, and you can see a wide range of products that we have developed. If you have any questions or products that you would like to discuss, please feel free to contact us. Thank you for your time and your attention.